So we talked a little bit about the DNA structure. We talked a little bit about the DNA structure. And we also started, correct me if I'm not mistaken, we started with the replication of the DNA. Did I? Or no?
Yeah. Yeah, we did replication.
Oh, yeah, we just started. We didn't finish it, right?
Yes. Okay, so we are going to catch up where we left last time for the DNA replication. Your exam scores are there. And folks, I was a little bit, I take that responsibility when it's my fault. We only have 3. The final exam is not comprehensive. We are not meeting on Monday because we are not meeting on Monday as such. About the fact that the DNA replication is semi-conservative, that means every time the DNA divides and it gets distributed to the daughter cells, only one strand of the DNA is distributed. So the daughter cell always receives one original strand of the bacterial DNA. And the other thing about the bacterial DNA is huh?
No, we can start here. Where you were.
We didn't finish the slide that you were on. We had like 3 more points at the bottom on that.
It is a very accurate process. Yeah, I was going to catch up on that part. So DNA replication is semi-conservative. We talked about the semi-conservative over here. This is a semi-conservative DNA replication. But every time a DNA divides, the daughter cells receive at least one of the original strands. Then we also talked about the fact that the DNA replication is asymmetric. That means one of the strand is always synthesized at a faster pace compared to the other strand. And that's why we have the leading strand versus the lagging strand of the DNA synthesis. We are going to talk about the leading strand and the lagging strand today in a little bit more detail, as well as the components of the DNA replication. DNA replication is a very accurate, DNA replication is bidirectional. We talked about it last time. And lastly, the DNA replication is a very accurate process because of the proofreading activity of the DNA polymerase. The DNA polymerase has a proofreading activity. So there's an editing activity. Every time it puts up a new nucleotide base, so if it is putting up a new nucleotide base against this A and T, G and C, T and A, and every time it puts up a complementary base, it makes sure that it is a correct base that is put in. If there's a mistake, it steps back, it removes the mismatched base, and then puts the correct one and then moves on. And that's why we say replication is a very accurate process because of due to the proofreading activity. Due to the proofreading activity of the DNA polymerase. So DNA polymerase, it maintains a high fidelity of replication. Any misincorporated bases are immediately removed, and it is the new correct base is incorporated in the DNA replication. Otherwise, just imagine what happens if the DNA replication doesn't have a proofreading activity. It is not an accurate process. Just imagine your skin cells which are being shedded and they are being replaced. Just imagine it like your nose cells will be growing on your skin. How creepy is that? Okay? Or some other cells will be growing on your face. That doesn't happen because the DNA replication is a very accurate process, and that accuracy is maintained because of the action of the DNA polymerase, which has a high fidelity and a good proofreading activity. DNA replication also requires energy. It's not a free ride. The DNA replication requires energy. And who provides the energy for DNA replication? Huh? Who provides the energy for the DNA replication? Look. Oh, don't guess what I write. Who provides the energy?
Isn't it just the cell itself?
The cell has to do it. I was expecting somebody would say ATP.
Oh, I said that.
You did?
But I said it really low because I'm not confident right now.
Oh no, I want you to be confident.
I'm
Thanks to
not.
that quiz.
But guess what?
There goes that. That's why I'm not confident.
That's why he was whispering, huh?
Hello, is that?
The one part is, you will think that ATP provides the energy for the DNA replication, but the truth is, ATP doesn't provide it. Every nucleotide that comes in to get incorporated, it comes as a DNTP. So it comes as a DNTP. So it comes in as CTP, deoxy-CTP, deoxy-GTP, deoxy-TTP, or deoxy-ATP. So whichever nucleotide needs to be incorporated, it brings its own energy. And that's why we tell it as NTP. That's why we have it here, DNTP, which is a generic name for the deoxynucleotide triphosphate. So every DNA, every nucleotide which is to be incorporated, it must bring its own energy. And that energy, which is being brought in by the nucleotide, it is always coming as like similar to the ATP. So when the CTP comes in or the GTP comes in, there are two of the high energy phosphate bonds are present. So here in this it is a DGTP. So there are two of the high energy phosphate bonds are present. When these are made free, it provides an energy. And that drives the G to form a phosphodiester bond with the 3' or with the 3' OH. So every time the DNA, which needs to be replicated and an incoming nucleotide comes in, every time it comes in, it comes as a nucleotide triphosphate, but then only this part. So this part is the only one with one of the phosphate group gets incorporated and it forms a phosphodiester bond. Sure. So every time a nucleotide comes in, it comes as a triphosphate. The last two phosphate group, because they are high energy phosphate like this or shown over here, this is the high energy phosphate bond. So these two bonds are broken and that provides an energy to form this phosphodiester bond between the 5th carbon atom and the 3rd OH group of the deoxyribose sugar. So this bonding is called as a phosphodiester bonding, and that's how the DNA chain elongation occurs.
Is that where the 5' and 3'?
That's where the 5' is always the blocked one. The new nucleotide always goes in the 3', 5', or 5', 3' because there's a OH group over here. At the 3', there's a carbon, there's an OH group, and the new incoming nucleotide forms the phosphodiester. The next part is replication initiates. It does not initiate randomly, but there are specific areas on the DNA from where the replication initiates. And that is called as the origin of replication. So DNA the replication process always starts not randomly, but from a defined area, which is called as the origin of replication. So DNA replication is not a random, the initiation is not a random thing, but it starts from a specific area, which is the origin of replication. And the replication process by itself is a collaborative effort of many components. Some of them are proteins, some of them are non-proteins, but they're all complex together as what we call it as replisome. Complex as replisome. The next slide talks about the replisome. What are those? Which is there in the next slide, and we are going to talk about that in a second. Now, the other thing about the bacterial DNA is one of the things which you need to know is the bacterial DNA is methylated. There's a methyl group that is added mainly to the C and the A groups. Mainly to the A as well as rarely to the C. So A is mostly, but C is rarely. They are methylated. That means they contain a methyl group in addition to all the other functional groups that are present within the DNA. And this methylation of the bacterial DNA has some significance because that methylated DNA portion is involved in the control of gene expression. Significance is, and it's in your textbook on page number 205. In some cases, some of the genes which are methylated can be turned on. Some of the genes which are methylated can be turned off. So bacterial gene expression. So that can basically control or regulate gene expression. It could be either turning on the gene or turning off the gene, depending upon whether they are methylated, the genes are methylated, or they are not methylated. They contain either the methylated adenine or the methylated C residues. At the same time, the methylation of DNA also is a starting point at the origin of replication where the initiation begins. Those areas are heavily methylated. So initiation of replication is also from those methylated DNAs, from a methylated DNA area. The third significance of the methylation is it allows the bacteria to degrade the viral DNA but do not disrupt the bacterial DNA. So it protects the bacterial DNA, but it would degrade the viral DNA. Anytime a virus goes inside, the bacteria might try to degrade the viral DNA because the viral DNAs are non-methylated compared to the bacterial DNA. So these are some of the significance of the methylation of the bacterial DNA.
Can you repeat the first one?
Yeah.
And the second one
Is the regulation of gene expression. Some genes can be turned on following methylation, some genes are turned off following methylation. And the viral DNA is non-methylated, so they are preferentially degraded. But the bacterial DNA, which is methylated, is protected. Degraded by the bacterial nucleases.
The bacterial
You said a gene can be turned on or off after methylation, like at the methylation point, that's when it...
Not after the methylation point, like some of the genes that contain the methylated sequence, those sometimes they are used to turn on the gene, and some of the genes can be turned off because of the methylation sequence. So regulation of the bacterial gene expression is mainly regulated by the methylation process. Now the next slide talks about the replisome, I believe, and we'll talk a little bit about the replisome. And in this context, we'll also talk about one of the antibiotics that is often used for controlling bacterial infection by targeting the DNA replication process. So replication, as I mentioned earlier, includes both protein as well as the non-protein components. The non-protein component includes the DNA template. So one strand of the DNA which needs to be replicated, that's a non-protein component. Then the RNA nucleotides. Because the RNA nucleotides are used as a primer, and I'm going to talk about those in the next picture, folks. What are the primers? And I'm going to catch it up because this is a primer of an RNA sequence. Energy, because energy is required for the DNA replication process. It's an energy requiring, what do you call it, as an anabolic reaction. It requires a deoxynucleoside triphosphate because those are the nucleotides that are incorporated in the DNA molecule. It also requires the participation of the origin of replication as well as the primers. So these are some of the non-protein components of the DNA replication process. What is the role of the primers and all? We are going to discuss this part, so don't get panicky, and I'll explain it to you to the best of my ability so that you are clear on that. So the protein components, these are the non-protein components which are here when I just mentioned. There are some protein component of the replisome that facilitates the replication of the DNA. And the replisome protein contains could be either with some enzyme activity associated with the protein, or they could be non-enzymatic. That has no enzyme activity, or there's no inbuilt enzyme activity associated with the two proteins. Those non-enzymatic protein includes the single-stranded DNA binding protein, which is also referred to as a stabilizing protein. It ensures that once the two strands of the DNA are separated, they don't snap back. As in this picture, if you look in this picture, these are the stabilizing protein. These are the stabilizing protein that binds to the separated strands of the DNA. You see that? So these are the stabilizing protein, or the single-stranded DNA binding protein. They prevent the hydrogen bonding between the separated strands. Then you also have the DNA polymerase. And I'm going to talk about DNA polymerase in a lot more detail in the next slide, where you have about what is the role of the DNA polymerase. So just bear with me for a second. Then you have the helicase. The role of the helicase is it helps to separate, helps to break the hydrogen bonds and separate the two strands. So the role of the helicase is breaks the hydrogen bonds between the strands and separates them. So the bond between the two, the hydrogen bond between the two strands, those are broken by the helicase, and it separates the two strands. In this picture, the next picture over here, in this picture, the helicase is the one that is breaking the hydrogen bond between the two strands, and so now the two strands get separated from each other. You also have the action of the gyrase. What the gyrase does, can I erase this part, folks? Did everybody write it? Are you? Did you write it? the gyrase, what it does, it alleviates the tension built up between the separated strands. It alleviates the tension buildup between the separated strands, and it does that by nicking one of the strands. By nicking. By nicking one strand. I think I did explain this thing to you a few days back in the lab. You remember with the rubber band?
Yeah. Oh yeah.
You remember? So that is the role of the DNA gyrase. And there are certain antibiotics which belongs to the ciprofloxacin group. It targets the DNA gyrase. So antibiotic like ciprofloxacin, it targets the DNA gyrase. So ciprofloxacin targets the DNA gyrase of the bacterial cells. Cipro is often used for the treatment specifically for the UTI, for the urinary tract infections. Cipro is one of those favored antibiotics, but it is never given to pregnant women because, so this is given for UTI treatment. And you know about the anthrax. Did I talk about the anthrax that day? That when we had that anthrax problem in Washington sometimes back, anthrax can be easily treated with penicillin antibiotics. So gram-positive bacteria, you can treat it with penicillin. But those individuals were treated with Cipro just because you don't want to take a chance and figure out later that they were using penicillin-resistant strains. So what they did was they treated it with ciprofloxacin. Those individuals were treated with ciprofloxacin, so it can be used for the treatment of anthrax, just in case you don't want to start treatment and then realize, oh my God, those were penicillin-resistant strains of bacteria. You need to be a little bit cautious in giving ciprofloxacin to pregnant men. Oh shit, what is that? Pregnant woman. Oh, it's not my day today. McConkey to McMonkey, pregnant men. So, it's never given to pregnant men. No, women. It is not given to pregnant women because there are some serious side effects. Because of some serious side effects, it really impacts the cartilage development of the newborn or the unborn child. That's one of the reasons why ciprofloxacin is never given to pregnant women, because it targets it, or it basically it has effects on cartilage development. So I'm trying to catch up on this one, so you know what is ciprofloxacin and what are some of the side effects, and it basically targets the DNA gyrase. Second of all, have a hairband or hair? Can I borrow?
Thank you. Yeah,
Borrow a hairband from someone, folks? Alrighty, folks, here it is. So the DNA, if you recall it from last time, is pretty supercoiled, and during the DNA replication, the two strands need to be separated like this. And the separation of the two strands is mediated by the DNA helicase. But there's always a tendency that the two strands will again come back together. Okay? Now, how would you prevent this from happening?
Cutting it.
So you cut it, and that cutting is done by the DNA gyrase. It cuts one of the strands. That's what is the nicking of the strand. Thank you. Now, the primase is the one that makes the RNA primer. So this one makes the RNA primer. Makes the RNA primer. Then you have the ligase, which joins the Okazaki fragments. Ligase is the one that joins the Okazaki fragments. Lastly, you have the RNase H. The RNase H, the RNase H is called as the RNase H because it only degrades the RNA portion of the RNA DNA hybrid. So RNase H, the H is called the hybrid of DNA and RNA, and it degrades the RNA portion. That's what is that's the RNase H protein. Before I go into the complete the play of the DNA replication process as it's shown in this picture, again, this picture looks a little bit scary, but I will try to make it as easy as possible, okay? So don't be scared out on this one. Before I get on to this one, I just want to catch up a little bit on the DNA polymerase, because the DNA polymerase is like the quarterback of the replication system. The DNA polymerase is one of the most important player of the DNA replication. And the DNA polymerase has certain properties. There are some specific properties associated with the DNA polymerase as such. It uses always one strand of the DNA to make the other strand. That's one thing which is universal. It always uses one strand as a template to make the complementary second strand of the DNA. It has a proofreading activity. That means there is some editing functions which are inbuilt within the DNA polymerase. It's something like this is a template of the DNA, and I'm the DNA polymerase, and I will incorporate, so look at this. This is the primer, because I cannot start from here. I cannot start from this point. There is an RNA primer here, which is complementary RNA sequence. I'm the DNA polymerase. What I do is I will put up the next complementary strand, and I will keep on extending the primer by adding complementary nucleotides. So I'll keep on extending the primer. And if I find, oops, I made a mistake last time. When instead of a C, I put a G here. So what it does, it backtracks, and that's where the editing role of the DNA polymerase is. It backtracks, it removes the misincorporated or mismatched nucleotide, replaces it with the correct nucleotide, and then it moves on. And that is what is the proofreading exonuclease activity.
So it's a dimer?
Huh?
A dimer that does it?
Not the dimer, is a, so this is the DNA strand. I'm putting up the new nucleotides here, and the new nucleotide, and I realize, oops, I made a mistake. I made some boo-boo in the last. So it takes a step back, removes that nucleotide, adds the correct nucleotides, and then again moves forward. You see that? So that's what is the proofreading. It's called as an exonuclease because it cannot go all the way down here and then realize, oh my God, I made a mistake there. You know, it doesn't do that. It just goes at the end. You know, that's why it is called as an exonuclease versus an endonuclease, where it can go anywhere in between. It elongates the DNA only in one direction, and that is always from the 5' to 3'. 5' to 3' direction, and that is the prime reason we have asymmetry in the DNA replication process. So it always extends the DNA in one direction, in the 5' to 3', and because of this activity, we have this asymmetry with the leading and the lagging strand in the DNA replication process. One strand is synthesized at a faster pace compared to the other strand, which is made at a slower rate. Not to mention, it can only add nucleotide. The DNA polymerase only adds nucleotide to an existing fragment of nucleic acid. And this existing fragment of nucleic acid is nothing but it's an RNA primer. So think about it, I'm the DNA polymerase, I keep on extending, this is the RNA primer, I keep on extending the RNA primer by adding complementary nucleotide to the DNA template. So that's my job, that's the job of the DNA polymer. But at the end, this primer needs to be removed because this is an RNA primer, and who removes that? Is the RNase H. Because this is a DNA template, this is the RNA primer. So RNase H will remove this RNA primer, and the DNA polymerase, one of the DNA polymerase will fill it up with the DNA nucleotides, and the DNA ligase will now connect the two. This whole sequence can be shown in the previous slide. But before I get on to that one, I just want to talk about the replication fork. This replication fork is the part where the active DNA replication is occurring. So once the DNA separates, this is where the new nucleotides are being added. This is the lagging strand. Then there will be a leading strand which will be chasing the fork, and that's where it's called as replication fork. And in the replication fork, obviously, the parental strands are no more paired. They are no more paired. And remember, in a replication bubble, which is shown here, in a replication, this whole thing is a replication bubble. In a replication bubble, there is one replication fork. They're going in two separate directions, but then they will come together. So this whole thing is a replication bubble. If you took it that way, this whole thing is called as a replication bubble. And in a replication bubble, there are two replication forks in the opposite direction. They go in the opposite direction, and there are two replication forks in the replication bubble where active DNA replication occurs. So with all of these, with this background, I think it's time for some little break. So when you come back from the break, we'll talk about, a few things about the antibiotics, the cell wall acting antibiotic and the sulfur drug and some of the ideal properties of antibiotics. So I would like to finish as much as possible, and if I'm not able to do it, we'll stop it there, okay? So, let me start from this one. So this is for the DNA replication. And all of these three pictures, because here it shows the initiation of the DNA replication. This picture shows the synthesis of the leading strand, and this part shows the synthesis of the lagging strand of the DNA. So initially, because the DNA replication process has these three steps, remember the DNA replication has the unwinding, the complementary base pairing, and lastly is the bond formation. Those are the three steps of the DNA replication process. Now, when you do the unwinding, that is mainly done by the DNA helicase. So initial process, these two get separated. The stabilizing protein binds to this one. It also prevents the reformation of the helix, is prevented by the stabilizing protein to a certain extent. Now, then comes the leading strand and the lagging strand. The leading strand is a continuous or an uninterrupted polymerization of the DNA nucleotides, and it's always trying to chase the fork. So it starts again from here. It has to first make up an RNA primer, which is the two of those things. What I put it over here, that's the RNA primer. And the DNA polymerase keeps on extending the RNA primer, as is shown over here. There is a 3' OH group over here, and the new nucleotides, which are coming as a nucleotide triphosphate, and the two phosphate groups are released. It provided energy so that not the hydrogen bond, then it can connect to this OH group over here, free OH group. So every time the leading strand is a continuous polymerization, and it is trying to chase this fork. It is always trying to chase this fork. It is moving in the 5' to 3' direction, and it starts from the origin of replication where you have this RNA primer, and it keeps on chasing the fork. It's an uninterrupted polymerization of the DNA nucleotides. That is the leading strand. The lagging strand is the one which is made on the opposite side, and it is a discontinuous DNA replication. It's a discontinuous because if it starts from here, and if it tries to do this way, then what happens is, by the time the fork comes here, this is starting from here, then what will happen to the other part of the DNA? So that was a kind of a big time question for quite some time. And Raji Okazaki, in his honor, we call this as Okazaki fragment, from MIT, he first saw, using a very high magnification microscope, he saw the DNA replication, and he found that there are two. One strand is being synthesized at a faster rate, which is the leading strand, and the second strand is synthesized in small pieces, and that's what we call it as the lagging strand of the DNA. And that is the time we also explain how the 5', 3' action of the DNA polymerase, how can it be satisfied in explaining the DNA replication process? He just passed away, just a few years back he passed away, and until the time he passed away, he was still a member of the MIT group of scientists. And in his honor, we call it as Okazaki fragment. Look at this, what happens. when it ends up, let me pull it down. Look, what happens is, this DNA, it opens up. This part is synthesized by the DNA polymerase, so this is the leading strand, is not a problem. Are you all okay with this leading strand? Now for the lagging strand, it starts from here with the RNA primer. So it provides a 3' OH and the DNA polymerase 3. That DNA polymerase 3 will be providing the nucleotide, and it will keep on extending up until this part. That's why the lagging strand is slower replication, because it is made in small pieces. Piece by piece by piece it is made. And here also, this is, look at this, RNA primer was made, and that was extended all the way up until here. And then this will be joined, once the RNA primer is released, then it will be joined to this segment by the DNA ligase. Okay? And again, when this comes all the way down here, this primer will be removed, and that will be filled in by the DNA polymerase, will replace the RNA primer with the DNA nucleotide, and then these two fragments will be joined by the DNA ligase. And because this is done in small segments, it is a slower process, and that's why we call it as the lagging strand of DNA synthesis. So there's a leading strand and there's a lagging strand. The lagging strand is the one that is done at a slower pace. It's a discontinuous replication. And Okazaki, there are many Okazaki. Here you see there are a lot of Okazaki fragments are being made. And all of these Okazaki fragments, like this is one Okazaki, this is second Okazaki, then they are all connected to each other by the DNA ligase. I don't want to tell you anything more on this, because I don't want to put up too much of pressure. I understand completely, folks, there are other things to be talking about. So I'll talk about the DNA replication at this step.
Wait a minute. I'm sorry. You said they're in between the DNA ligase, but it looks like they're in between the
What is it?
It looks like they're actually in between the RNA primer.
So here is what happened. The RNA primer has to be removed by the RNase H.
Right.
Because in the DNA you cannot have an RNA sitting in there.
Gotcha.
So what happened? The RNA primer will be removed, and that gap, these three nucleotides need to be filled by the DNA polymerase 1.
So the RNA primer is just a fill-in.
RNA primer needs to be filled in, and that is taken care of by the DNA polymerase 1.
Gotcha.
And DNA polymerase, like this is a template. If I'm the DNA polymerase, I cannot start from here. I just cannot start from here. Are we good?
I remember the Northwest earthquake in 4th grade, so this is like nothing. Oh, I can't stand it.
You made me scared, like I was gonna die. Okay. But what about the aftershock?
Can we go home now?
Oh my God.
Oh, you guys, the building's gonna collapse on y'all. I thought it was in my pocket. Oh my God.
We can be more physical next time an earthquake comes around. Just push me. You're like, get out the way.
I didn't mean
I know.
No, you don't.
How do you also
So, I am not from California. This is still not.
two of the processes. One is the transcription.
Wait, you still didn't get fill in the blanks for polymerase.
We don't need to know that. It's not going to be on the test. How about that?
The answer to my question
I just like that. Because we can.
It includes the transcription and the translation process. And these are two separate processes, but they're interdependent in the sense translation cannot occur until transcription is done, and once the transcription is done, translation is inevitable. So these are two independent but separate processes, and during the process of transcription, an mRNA is synthesized. During this process, mRNA is made. You don't have to know these ones. This one you don't know. An mRNA is made in the process of transcription, and the DNA acts as a template. So from the DNA, the RNA is made, and that is the process of transcription. And translation is when that mRNA is to be coded into the protein sequence, a protein amino acid sequence. It is to be the amino acid sequence of proteins. So that is the translation, and this part is shown over here in a much better way. So this is the DNA. From DNA, then it's not that the DNA is converted, but the DNA, one strand of the DNA is copied. That is the most important word. DNA is copied to the RNA sequence in the transcription process. And once it is transcribed, once the RNA is made, invariably it will be translated by the ribosome to the amino acid sequence of proteins. You have to go back because this transcription process is done by the RNA polymerase. And RNA polymerase is different than the DNA polymerase in the sense the RNA polymerase has very poor proofreading activity. It has a very poor proofreading activity, and at the same time, the RNA polymerase, which makes the RNA from the DNA template, it also makes it in the 5' to 3' direction. And in the RNA there is, in the RNA per se, the one which is the product of transcription, the RNA is always single-stranded. It's not a double-stranded RNA. It is always single-stranded. It is not a double-stranded. That's one thing. There are 4 types of the nucleotides. Oh, I'm sorry. There are 4 types of the RNA are being made. Those 4 types includes the messenger RNA, the transfer RNA, the ribosomal RNA, and the primer RNA.
Sorry, can you repeat the T and R?
The mRNA, tRNA, rRNA, and the primer RNA, and these three are involved in the translation.
What's rRNA again?
Ribosomal RNA.
And T?
Transfer RNA. So ribosomal RNA, transfer RNA, and the messenger RNA, they are involved in some or the other way in protein synthesis. Whereas this one, the primer RNA is involved in the DNA replication. This is involved in the DNA replication. You still have the purines and pyrimidine, but there is no T. There is no thymine residue in the RNA. In RNA, you have just the ribose sugars. There is no deoxyribose. In the RNA, you have the ribose sugars, but not the deoxyribose. Okay? I will stop it here, folks, for today. We'll see.